In this lecture, we're going to take a look at the structure and function of DNA and how it's replicated and translated into proteins that the cell can use. So back even just less than 100 years ago, back in the 1940s, we had this idea that chromosomes, what we could see in the nucleus of the cell, consisted of DNA and protein, but we weren't sure exactly which was the actual genetic material. Was it the DNA or the protein? So even back in the 1940s and 50s, we still weren't exactly sure how the cell could make all of its stuff and function and all that. So it was in the 1950s that Hershey and Chase demonstrated that it was actually the DNA part of the chromosomes, not the proteins that were in the chromosomes, that was the actual genetic material, the thing that was passed down from generation to generation and was responsible for like the instructions for all of the cell's workings and such. So this kind of goes to show that this our understanding of DNA is actually fairly new in terms of historical time and stuff like that. So now that we know that it's the DNA that's the genetic material that is going to be the instruction manual for the cell, let's take a look at the structure of DNA and its partner in crime, RNA. So we talked about these very briefly when we talked about the molecules of life, the carbohydrates, lipids, proteins, and nucleic acids. Well, DNA and RNA are the nucleic acids, right? They are very long chains that are made up of the monomer nucleotides. Okay, the nucleotides are going to have three parts. We've got a phosphate group, okay, a 5-carbon sugar, and a nitrogenous base. We're going to see that the 5-carbon sugar is one of the things that's going to be different between DNA and RNA. DNA has a sugar called deoxyribose, and RNA is made up of the sugar ribose. And so deoxyribose just means that we are missing an oxygen from that sugar. Okay, so there's 4 different nitrogenous bases, okay, that exist in DNA. And these nucleotides that are made up of those 3 parts are always covalently bonded between the sugars and the phosphates. So we can see in the picture below that this part is the phosphate and this part is the sugar, and we've got a covalent bond kind of between the phosphates and the sugars. Okay, so we're going to see that's we call a sugar phosphate backbone. Let's see, like we have here, it says both DNA and RNA have a sugar phosphate backbone because they are connected to each other through those phosphate and sugar groups. Now the nitrogenous bases are kind of like little arms or appendages along the backbone. So if we take a look and we say, this is the backbone of it, we've got a bunch of these little nitrogenous bases that kind of stick out, okay, from it, like little arms. Now, bases can come in 2 types. We can have the single ring structures of thymine and cytosine. So those are our T and our C as our single ring. So that's like, you can't quite see it behind it, but that's like this. There's only one ring behind there. So our C and our T are single ring structures, and then we also have double ring structures as well, and that's our adenine and guanine, A and G. And so the adenine and guanine, the double rings, we can see kind of popping around here, that there's 2 little rings right there. So that is our adenine and our guanine that are going to be double ring structures. Now RNA is slightly different. Instead of thymine, so instead of the T, we have a uracil. Okay, so we have a U instead of a T in RNA. And like I said before, we have a different sugar called ribose versus deoxyribose. But otherwise, the other characteristics are pretty much the same between DNA and RNA. In the 1950s, Watson and Crick actually discovered the structure of DNA. So they, through X-ray crystallography, I believe, figured out that the sugar phosphate backbone was on the outside of the structure of DNA, and the nitrogenous bases were all kind of on the inside. And there was hydrogen bonding between the nitrogenous bases to keep those two strands together. And what he saw, or what they saw, I should say, was that it was always the A's and the T's hooking up, okay, or hydrogen bonding together, and the G's and the C's hydrogen bonding together. Never saw any other types of combinations. So an A and G weren't ever bonded together, or a C and the T weren't ever bonded together. It was always A with T and G with C. So if we look at the right-hand side here on the pictures, we've kind of got varying different models here. This is the simplest one here in the middle, where we can see A and T hooked up together by hydrogen bonds, and G and C hooked up together by hydrogen bonding, 3 hydrogen bonds there. This would be kind of like what it would look like in the space filling model, and this is our double helix structure, okay, that they coined after they figured out that it was 2 sugar phosphate backbones together with the nitrogenous bases in the middle being hooked together with a hydrogen bond. So they coined the structure of DNA as what we call a double helix. Okay, so it's 2 strands of the sugar phosphate backbone, and it's twisted together, kind of in this coil type of a structure. And the 2 strands are going to run in opposite directions. Okay, so if we think of one strand having one end being what we call the 5 prime, and the other end being what we call the 3 prime. Okay, so if this strand right here went like this, okay, then the opposite strand, okay, would have the 5 prime down here, the 5 prime end down here, and the 3 prime end up here. And so they run in opposite directions. And that's going to be important when we talk about DNA replication and how it copies itself. So see, the structure is going to suggest the method of replication because the structure suggests that these 2 strands could come apart and serve as a template for copying, the complementary strand using, the same base pairing rules, the A's with the T's and the G's with the C's. Okay, so what happens is that this double strand has to come apart first, and then we could get little, protein machinery to come in and kind of copy the, what the strands here, okay, individually, okay, and we can get what this orange strand is the new complementary DNA that is going to be not identical to what it copied, but the complement of it. This mechanism of DNA replication seems, pretty simple when you're explaining it or thinking about it. The 2 strands come apart, and then we make the complement strand as, a new strand of DNA. Bam. Okay. However, it's actually really complex when we look at it. We need dozens of enzymes and other proteins in order to do this. In fact, only the one that we're actually going to focus on is what we call DNA polymerase. And that's the main one that's going to join nucleotides that are inside of the cell together to form new sugar phosphate bonds that are complementary to the original DNA sequence. Okay, so DNA polymerase is the main enzyme that we're going to focus on in DNA replication, because that's going to do kind of the main job of putting together, it kind of has a bag full of nucleotides that, as it goes along the DNA strand says, okay, well, there's a G, I need a C. Bam. Let's kind of hook that onto the string. The next one is an A. Okay, I need to, pull a T out of my bag and hook it on and make that bond between the 2 nucleotides. So that's kind of the main job of DNA polymerase. It also has the job of repairing damaged DNA, right? So if a bond is, broken, or, kind of the little nitrogenous base is kind of hanging off, by one strand or something, it's going to come in and be able to fix any part of the DNA that needs to be fixed. So it also has to do not only with DNA replication, but with also repairing damaged DNA as well. Now, when we talk about DNA replication, there are many different sites along the DNA, because the DNA strand is very long. So instead of just starting at one end and going down, we've got multiple origins of replication. Okay, so a bunch of little DNA polymerases will hook up at different locations along the DNA and start replicating. So it'll pull the DNA apart and make what we call kind of replication bubbles. Okay, it could, kind of look like this. So it pulls it apart and then starts to copy. And as you can imagine, as it goes along, right, then bam, once they meet each other, then that part of the DNA is done. It can unhook itself and go to a different part of the DNA. So in the end, we get two daughter DNA molecules. One strand of the double helix will be kind of the what we call original DNA that was there, and then the other strand is the new complementary DNA that makes up that double helix. So that's how we get two daughter DNA molecules. And like I had mentioned, there's many origins of replication that exist along a strand of DNA or a chromosome. This is going to greatly shorten the time required for replication, because we can start at many points and get little bits and pieces done, rather than having to start at one end and unzipping the whole thing, and just having one machine go down the entire length of DNA. And then, just like I had mentioned, we get two daughter DNA molecules, one old, one quote unquote old, and the other one is the new complementary strand that was just put together by the DNA polymerase. Okay, so that is basically how DNA is going to copy itself or replicate itself, and that was on the very, very simple end of an explanation of how it's done. Like I said, it's super complex with dozens and dozens of enzymes and proteins that are involved, and each have their own name and function and stuff like that. But at this level of a course, we don't need to get into all of those specifics. Now we're going to kind of shift gears and move on to how is DNA then translated into proteins and characteristics of an organism. So if you remember back in the genetics chapter, we talked about genotype, which is the sequencing of the DNA. And then we get what's called a phenotype, or what the organism actually looks like and how it works. So it's the organism's specific traits that are visible and invisible as well, like working on the inside rather than everything we can see on the outside. So the molecular basis of getting a phenotype is going to lie in the proteins. Okay, so that's super important. It's the proteins that is made from the DNA. And these proteins are going to have different functions, and they're going to function differently in different organisms, and that's why we've got different phenotypes. All right, so the kind of the central dogma of molecular biology is this idea that DNA goes to RNA, and then it's translated to proteins. Okay, so we've got DNA is going to specify the synthesis of proteins, but in an indirect kind of way, because DNA proteins aren't actually made directly from DNA. DNA first sends a copy of RNA into the cell that is then translated to proteins. So RNA is going to program protein synthesis, and DNA is going to send out kind of the instructions via this RNA form. So we have DNA in the nucleus that sends out just specific parts of itself as instructions manuals for RNA, and then this RNA is translated into proteins outside of the nucleus. Okay, so these two main processes have their own names. Transcription is going from DNA to RNA, okay? And then we have translation that goes from RNA to protein. Okay, so transcription is when DNA copies itself into RNA, and translation is when RNA translates that sequence into actual functional protein within the cell. All right, this transcription and translation process is pretty intense. We're going to take a look at an overview first before we hash out all of the details between the two. So the DNA language is basically kind of in the form of those 4 nucleotides, and they're going to be in specific alternating sequences depending on what proteins eventually have to be made from that information. Now RNA has that same kind of language of those 4 nucleotides. So the transcription of DNA is going to give RNA with the same nucleotide language. Okay, so DNA and RNA kind of speak the same language. We've got the same type of nitrogenous bases in DNA as we have in RNA. Now, the nucleotide bases of RNA are going to be complementary to DNA, right? They're not going to be exactly the same. So if we take a look at this sequence here on the right, if DNA has an A, right, and adenosine, then RNA is going to hook that up with a U. Okay, so it's complementary. It is not an exact copy, but it's complementary of it. And this complementary bases are then going to be used in order to translate that to proteins. Now, unfortunately, the proteins use a different language than DNA and RNA, so we need a translator. So that's kind of basically why it's called translation when you go from RNA to proteins. Okay, so remember, proteins are made up of polypeptide chain, and the monomers of proteins, or those polypeptide chains, are the 20 amino acids, okay, that are going to kind of form in whatever sequence that the DNA tells it to. Okay, so this polypeptide chain is, again, those 20 amino acids. This is different than the four nucleotide bases that we have in RNA. So we're going to see how this translation occurs as we take a look at the process. Now, RNA, remember, is just the messenger, okay, from DNA. So DNA is the instruction manual. RNA is just the messenger that tells how to make the proteins. To get from a RNA strand to a strand of actual polypeptides or proteins, what needs to happen is that for every 3 nucleotides on the RNA molecule, okay, we call that a codon. Okay, triplet base pair is what we call it. So this triplet base pair is called a codon. Each codon is going to have a specific amino acid associated with it. So 3 base codons in the DNA transcribed into 3 base complementary codons in RNA, which translate into an amino acid within the protein chain. Okay, so for example, in DNA, the 3 base codon is AAA. Okay, in RNA, we get that translated into UUU. Okay, and we're going to see that it's the UUU is going to be the code for a specific amino acid within the polypeptide chain. And of course, we humans have kind of figured out this genetic code through research. So somebody has figured out, which codons translate to which specific amino acids, and they put it in this handy dandy little chart here. So we start out with the first base on the left. So if your first base is U, Okay, then you take a look at the top, okay, and take a look, okay, what if the second base was C? Okay, so we're going to look kind of in this area here, and then the third base is on the right hand side, so we take a look within each of the individual boxes. So if it were G, okay, we take a look right here, UCG, and that's going to code for the amino acid serine. Okay, so you can see that we can have multiple codes for the same protein. Okay, so we have some, kind of redundancy a little bit, but there's not really kind of any ambiguity. So this kind of is actually a good thing because, say if there's a mistake in, a codon of the DNA. So, if we had a UC and it's supposed to be G, but what if it got translated as UCC? Okay, we still want the serine amino acid, but we don't, if UCC was a completely different amino acid than UCG, then we'd be kind of screwed with our protein. Okay, so we have kind of a little bit of wiggle room when it comes to decoding the DNA into proteins, which is a good thing. So 61 of the 64 codon kind of possibilities are going to be used for making amino acids. And then there are 3 stop signals. Okay, so we've got these guys over here that are stop signals, UAA, UAG, and UGA. Those guys mean stop making the protein, we're done with it. Okay, and then we've got 1 start signal. So anytime it's a code for UAG, it's methionine, and that always is going to start the protein sequence. Okay, so every protein is always started with the amino acid methionine. So let's take a look at the nitty-gritties. That was the overview. Let's take a look exactly and see what happens at the detailed parts. So in transcription, remember transcription is going from DNA to RNA. Okay, the first thing that has to be done is that DNA strands need to unwind. They need to break apart so that RNA can be made from one of those strands. So we've got RNA polymerase, that is the key enzyme here to make RNA. It's going to come in and start to unwind part of the DNA and make a complementary copy of that DNA strand. All right, so only 1 strand of DNA is transcribed, and this transcription member is a complementary copy of the DNA strand that it's using. All right, so remember that means that if the DNA is a T, then the RNA is an A, okay, because A always hooks up with T, okay, and then, the next one, if it's an A, then in RNA, it's going to be translated as a U, because remember, there's no T's in RNA, there's no thiamines in RNA, it's been replaced by a uracil nitrogen base. So this RNA polymerase is going to basically go along the DNA and read the code and put together our new strand of RNA as it goes along. And if you take a look, the new strand of RNA is going to be kind of the exact copy of the strand of DNA that's not being translated, okay, or transcribed, I should say. So see here, this A is like that A, and then the U is the T, and there's C, C, A, T. So we get, even though the RNA is a complementary code of the 1 strand of DNA that it's actually copying, it's going to be the exact copy of the other strand of DNA. So we're not going to lose any genetic information along the way. Okay, so how does RNA polymerase, which is the main protein, remember, that we're going to talk about in transcription, how does it know, where to go on the DNA? Well, each kind of part of the gene in DNA has what we call a start signal. It's what we call the promoter, is the start signal. And so RNA polymerase will just kind of go down the strand and look for start signals. Okay, so that's where the RNA polymerase is going to attach to. They're going to attach to these promoters or the start signals along the DNA. All right, the first phase of transcription is what we call initiation, the beginning, and that's the attachment of RNA polymerase to the promoter site and the start of the synthesis process. And remember, the promoter is going to dictate which strand is transcribed, whether it's the 5 to 3 or the 3 to 5 one. The promoter will tell the RNA polymerase which one to copy. The second phase is what we call elongation, okay, because as RNA polymerase goes down, unwinds the DNA, and starts making a copy of the specific DNA strand, an RNA molecule is going to start to grow or elongate. Okay, so that's why it's called elongation. The DNA region that's already transcribed then kind of winds back together and returns to its double-stranded structure. So after RNA polymerase is done with that particular strand of DNA, the DNA is going to wind back together and become kind of, brand new again. All right, and the third and final phase of transcription is what we call termination. When the RNA polymerase reaches a special sequence of DNA bases called the terminator, okay, or the stop signal, then the RNA polymerase, the protein there that's making the RNA, is going to detach itself from the DNA, and the RNA molecule will separate from that RNA polymerase protein and go out of the nucleus to send that message to make that particular protein. In eukaryotic cells that have a nucleus, this transcription, these 3 phases of transcription, will all occur inside of the nucleus. And so this newly synthesized RNA molecule has to go out into the cytoplasm in order to be made into a protein. Okay, so what happens is that before that RNA can go out into the cytoplasm, it has to be kind of modified first. It has to be, kind of protected in a way. So let's take a look at those modifications. Basically, what has to happen is that inside of the nucleus, the RNA gets caps and tails, which are extra sequences on the ends of the RNA protein that protect it from degradation while it's outside in the cytoplasm. Just in case we've got some lysosomal enzymes or whatever that come and want to try to eat it up. Those caps and tails are gonna protect it. And those caps and tails also are going to indicate to ribosomes that, hey, this is a messenger RNA molecule that needs to get translated into a protein. So they're also kind of an indication that, hey, here's some instruction manuals coming, so you ribosomes can start making some proteins. So caps and tails are one type of modification that the RNA molecule undergoes before it gets out into the cytoplasm. Another is what we call splicing. Now, RNA molecules are going to contain coding and non-coding sequences within itself. Okay, so if we take a look at this example, right, we have EXA and then this junk in the middle and then MPLE. Okay, so we have to cut out the junk in the middle in order to get our word example. Now, the non-coding sequences, the ones that are junk, okay, are called introns. The coding sequences, the ones that we want to keep for particular proteins, are called exons. All right, so we need to remove the introns from RNA molecule and put together all the exons of the RNA molecule. And so that process we call splicing. RNA splicing. We take out the introns and we splice together those exons to make the complete sequence for a particular protein. Now, RNA splicing is going to allow the production of more than one polypeptide from an RNA molecule, right, by varying the splice points. So we take out certain points, certain introns here and there, and splice the exons together, we can get one type of polypeptide chain, but we can also take that same RNA molecule and take out different points and splice together different exons to get a different polypeptide molecule. So it is completely amazing how your cell can actually do this and make sense of all of this stuff. All right, so after we've capped it, tailed it, and spliced it, now the messenger RNA is ready for translation. Okay, so we call this coming, the one that's transcribed from the DNA into RNA, we call this mRNA, messenger RNA. Okay, because it's going to be the messenger that goes out into the cytoplasm to find ribosomes to translate it. Okay, so translation occurs in the cytoplasm, so the RNA needs to exit the nucleus through those nuclear pores. And we're going to take a look at the other components for translation that are also in the cytoplasm. All right, so now we've moved on to the translation part. Remember, transcription is going from DNA to RNA. Once the RNA is all ready to go, we're going to send it out into the cytoplasm, and translation is going to occur. So these are the things that are needed for translation. The mRNA, the messenger RNA that came from the nucleus. We also need ATP, and we need ribosomes, and we need amino acids, and we need what's called transfer RNA or tRNA. Transfer RNA is kind of basically the molecular interpreter that's going to interpret the three codons of the three base pairs in the messenger RNA into the language of proteins, which are the amino acids in the polypeptide chain. So transfer RNA is like our translator RNA that's going to interpret the three-letter code from the mRNA into the one amino acid for the polypeptide chain. All right, so the amino acids that are going to make up the polypeptide chain, they're kind of all floating in the cytoplasm, but they're like, blind mice. They can't recognize the codons in the mRNA directly, so they need tRNA to help them. So tRNA molecules have basically on one end they attach a specific amino acid to themselves, and they bring that amino acid over to the ribosome for protein synthesis. So we're going to have tRNA that matches an amino acid to the appropriate codon in the mRNA. So if we take a look at the picture on the right, this is kind of a picture of over here would be like a tRNA. So on this end of the tRNA, we're going to have an amino acid. Okay, so he kind of has one side of him that sticks to amino acid, and the other side of him, we have what's called an anticodon, which is going to read the mRNA. Okay, so if its anticodon matches up with the codon on the mRNA, it's going to then attach that amino acid together with the previous one. So we have two basic functions for this tRNA. One end is picking up the appropriate amino acid, and the other end is recognizing the appropriate codon in the mRNA, right? So remember how I just said that at the one end of the tRNA we have a special triplet of bases called an anticodon, okay, which is going to match up with the actual codon from the mRNA in the sequence. This anticodon is complementary to the codon in the mRNA. Okay, so it binds using those base pairing rules, right? Remember, A with U, G with C. And again, it's going to bring that specific amino acid to be added to the polypeptide chain within the ribosome. So here's basically kind of where the workshop where the polypeptide amino acid chains are being hooked together, and they're called ribosomes. Okay, ribosomes are found in the cytoplasm, or they can also be found on the rough endoplasmic reticulum. They consist of two subunits, a small subunit down here and a large subunit on top. Okay, each subunit is going to consist of a large RNA called ribosomal RNA, okay, and lots of different proteins inside to hook the amino acids together. So let's not confuse ourselves with all the different kinds of RNA. We've got messenger RNA, right, that is made from the DNA. Okay, so that's from DNA. We have tRNA, that's going to bring the amino acids. And we have R ribosomal RNA, rRNA, okay, that makes up the ribosomes, or is in the ribosomes. So we've got different types of RNA molecules that have different functions. All right, so the small subunit of the ribosome, this guy down here, is going to have an mRNA binding site. So this is kind of where the mRNA is going to feed through, okay, with its codon, or its little codons up here. And then the large subunit has two binding sites, one called the P site and one called the A site. Okay, so if we take a look, here's the P site, it's the same as this guy over here, and here's the A site. Okay. Now the P site is going to be holding the tRNA that has the growing polypeptide chain on it. Okay, so we can see that here's the growing polypeptide chain, and it's at the P site. The A site holds the tRNA that carries the next amino acid. Okay, so this guy is the next amino acid that's going to be hooked up to the polypeptide chain. So it's kind of like the P site is batter's box, the A site is the guy on deck. Of course, just like transcription, translation also has three phases. Okay, so our first phase is initiation. In initiation, we're going to bring all of these components together that we need in order to translate the mRNA into a protein. Remember, the mRNA is longer than the genetic code that it carries, because it has those caps and those tails that are going to, basically help the mRNA hook up to ribosomes and get where it needs to go. During initiation, we're going to determine exactly where the translation is going to begin. So if we take a look at the picture on the right, we can see that this strand right there is the mRNA. The light pink is likely the cap. That kind of is basically saying, hey, this is an mRNA, I need a ribosome to help translate this. So it's kind of the beginning for the binding of the mRNA to the ribosome. And then we have the dark red, which is going to be the actual genetic sequence that's going to be translated. So we're going to have to get the first tRNA there in order to start that translation. So the first tRNA is always a methionine. Remember, it's always going to be AUG as the start codon for the mRNA. So we're always going to have methionine as the first polypeptide in a sequence. So this tRNA that's going to hold the methionine is going to enter the P site on the large ribosome subunit. Okay, and so we've got the mRNA down here, okay, that is attached to the small subunit, and then that initiator tRNA that carries the methionine amino acid is going to sit at that P site. Once we get all of that kind of together, that's our initiation, and the initiation phase is complete. Once we've got the small subunit hooked up with the RNA, the mRNA, And then we've got the large subunit there with the first initiator tRNA in that P site. Initiation is complete. So we can move on to the second phase, which, again, kind of just like transcription, is called elongation, because the next tRNA carrying the amino acid is going to come and bind to the A site in the large subunit. And remember, it's going to have base pairs that are going to be complementary to the mRNA that's sitting in the small subunit. Okay, so if we take a look at the picture, here we have, basically this is the mRNA. It's got the little codons, and each three are going to be read by a certain tRNA that carries the specific amino acid to the A site. And then we get the polypeptide dehydration synthesis there to make the polypeptide chain. And that the tRNA that's in the P spot is going to be let go of, and then the one that was in the A spot kind of shifts over and allows, again, another tRNA, the next tRNA, to come in to hook up its amino acid to the polypeptide chain. Okay. So that's kind of basically the overview of the elongation process, and this keeps going and going and going until we're done. Okay, so this kind of first part here, where the tRNA comes in and sits in the A site, is called the codon recognition step, because it's got to have an anticodon that recognizes the codon within the mRNA that's complementary to each other. Okay, basically just how I explained. Once this tRNA gets there, we're going to get the second step of elongation, which is the polypeptide formation. Okay, and then the polypeptide in the pRNA is going to leave, or the tRNA within the P site is going to leave, and the one from the A site goes over to the P site, so it kind of shifts over, allowing more room for the next tRNA to come. Which is basically what all of this information on this slide says. This step here where it shifts over is called translocation, where the P site tRNA leaves the ribosome and the A site RNA moves over to the P site, allowing another tRNA to come into the A site to bring its amino acid to hook up with the polypeptide chain. And this happens over and over and over again, and it repeats until we get to the stop codon. And this is the third and final phase of translation, which is called termination. Right? So when elongation will continue until the stop codon appears on the mRNA in the A site, and then translation stops. The polypeptide is freed, the ribosome subunits come apart, and there's no more translation. All right, so this is a great summary picture that you're going to need to know the details for. It's looks like figure number 10.20 in your chapter text. So if you can't quite read everything on this slide, please make sure you go back to your textbook and copy this picture down. This is basically, in picture-wise, what we said in words in the last 20 slides. So this is a great review. You're going to need to know these details, how this works. You might even have to draw it and explain it in your test, in your midterm coming up, hint, hint. So make sure you know how this works. Okay, so that was the end of transcription and translation. The last little section we're going to include in this lecture is called mutations. So this can happen. Mutations are any changes in the nucleotide sequence of DNA, and it can involve either very large regions of the chromosome, excuse me, that were mistranslated, or it could just be a single base pair here and there that gets mistranslated and therefore changes the protein, which then changes the phenotype. So that's actually one of the things that happens in sickle cell anemia, is that it's just a simple kind of mutation of one single nucleotide that changes one of the amino acids in the polypeptide chain, but it throws the entire protein off. So in mutations, there are basically two different types. We can either have base substitutions, or we can have insertions or deletions. In base substitutions, what happens is that instead of the correct base being hooked up in the mRNA, we're going to have a different base. So if this was the normal gene, okay, and we have this base here that's supposed to be a G because it's going to code for the amino acid glycine. Instead, what if we have a mistake and now we've got A here instead of G, and so AGC codes for serine, okay, which is a different amino acid than glycine. So we've got this substitution here. Now this can either not affect it at all, okay, so whether there's a glycine there or a serine doesn't matter. Within the polypeptide chain, it's not going to make a big difference. So we can either see no change, or we can see an insignificant change. So some base pairs, substitutions, remember, we've got that little wiggle room. Looking back at the genetic code, we do have a little wiggle room there with the second and third base pairs within a codon. So sometimes we can either see no change or a very insignificant change, meaning that we're going to get the same amino acid coded anyways. Or we can get a completely different, drastic change where we've got a hugely different amino acid being coded now that's going to change, the primary, secondary, and quaternary and tertiary structures of these amino acids and totally change their function. So it's just kind of depends on where the substitution takes place within a codon. Now the second type, the insertions or deletions of one or more nucleotides, can actually change what we call the reading frame of the mRNA. It can change it into entirely different protein and protein sequence, unfortunately. So this is going to likely, basically make a non-functional protein. So if this were the normal gene up here, we've got a base deletion. So we, for some reason, this U was deleted from the sequence. Now, instead of UUU, which is phenylalanine, we get UUG, which is leucine, completely different amino acid, and also changes all the rest of the amino acids because it shifts the reading frame, right? So now this G right here was supposed to be the first G for glycine, but now it's the ending for leucine, and so if we have this second, GCG is going to be for alanine, okay, instead of glycine, which we should have had a GGC. Okay, so we can get totally different amino acids if there's insertions or deletions within the mRNA sequence. Most of the time, mutations are going to lead to non-functional stuff or very different, changes and harmful things. But some mutations can lead to useful adaptations and, different diversities of organisms, different colors and stuff like that. So mutagens are physical and chemical agents that basically cause the mutations within genes or within transcription. So high energy radiation, which is used sometimes to battle cancers and tumors, is basically, they beam a high energy radiation waves at the tumor part of your body, and basically what they want it to do is cause mutations in the DNA so that the cancer cells are no longer cancerous and die. We can also have chemicals in our environment that can cause mutations in the DNA, and those are called carcinogens. But sometimes mutations happen just as simple errors during replication of DNA. Okay, so sometimes DNA polymerase during replication is just going to make an error. Okay, it happens. Same thing in transcription. Sometimes the RNA polymerase just, again, makes an error. And we can get a mutation from that. Luckily, though, like I was saying when we were talking about the genetic code, there's a little bit of wiggle room. So we've got the redundancy within the genetic code and the codons, where sometimes the second and the third base pair doesn't really matter what it is; it's still going to code for the same amino acid. So that actually kind of helps decrease the effect of any mutations. So that's kind of good in a way. Remember, we can have GGC, GGA, GGG, and that all is going to code for glycine. So we can have a little wiggle room there, which is going to help decrease the effect of mutations. Now, if we do have a mutation that changes an amino acid for another, we call that a missense mutation. Okay, so again, here instead of glycine, we get serine. So that's what we call a missense. Now, sometimes the mutation can change the coding from, it's supposed to code for a specific amino acid, but instead it changes it to a stop codon. Okay, and in this case, the translation is going to end early, right? We're going to get a protein that gets cut off halfway in between. So this is what we call a nonsense mutation, because we don't get the complete protein in the end. All right, so that kind of concludes our lecture on DNA structure, function, replication, transcription, and translation. So I'm going to want to make sure that you go back to your textbook, get the appropriate pictures, information, and kind of work through this and get this, different parts of each of these things in your head, and make sure you know the difference between the initiation part in transcription versus initiation in translation, and all of those different details.